Elisa Boscolo. Elisa is an associate professor at Cincinnati Children's Hospital. Prior to going to Cincinnati Children's Hospital, she worked with Joyce Bischoff, and in Joyce's lab, she made several monumental discoveries on infantile hemangioma. And since forming her own group, she's continued to do amazing work on vascular malformations, such as venous malformations, and developing a really nice xenograft model and doing amazing preclinical studies, not only looking at rapamycin, but also new therapies as well, which has led to really nice publications. She's also worked on combined malformations, and today we're going to hear more about GNAQ mutations and how they result in vascular malformations. So not only is Elisa a great scientist, she's also a great friend, and I always enjoy having a drink with her at meetings and hearing about the great work that her lab is doing. So with that, I'll turn it over to Elisa for her to start her presentation.
Thank you so much, Mike, and thanks for the LGDA for always having me as a guest or to inviting me to present. And so today I want to tell you about a story that we published last year on vascular tumors with GNAQ mutations and what we have done so far about that. And okay, so before I move on, I really want to sort of specify that what we're working on at this point are vascular tumors with the mutation in GNAQ or GNA11 and 14, which are highly homologous genes. And I want to sort of clarify that there are two types of vascular anomalies that are associated with GNAQ mutations, which are vascular tumors on one side and vascular malformations, namely capillary malformations. And the difference between those type of mutations and type of disease is that there's, so ones are vascular tumors, ones are malformations, and that the type of mutation is different. In vascular tumors, we see this mutation Q209, and the most frequent substitution is Q2 from Q to L. And in capillary malformations, the mutation is at the site R183. And so the focus of my talk will be the mutation Q209L. Okay. So the type of vascular tumors that are associated with somatic hyperactive GNAQ mutation at the site Q209L or conserved site in GNA14 Q205L are congenital hemangioma, for which work from Matt Warman's lab have found GNAQ mutations associated with about 80% of the cases they looked at. And then we have a handful of cases of pyogenic granuloma, caposiform hemangiotelioma, and tufted angioma. And I want to say that for those, again, those are handful cases, but for caposiform hemangiotelioma and tufted angioma, we don't have many other mutated genes that have been associated with them. So that's kind of a way to start, you know, looking, investigating those diseases. And although there are some clinical therapies, namely corticosteroids and type of chemotherapeutics for vascular tumors that are in use, there are really no FDA-approved targeted therapies that are specific for these patients. And so before I move on to tell you a little bit more of what we're doing, I want to introduce to you the G-alpha-Q. So the G-alpha-Q is the name of the protein, and the gene name is GNAQ. That stands for guanine nucleotide binding protein, and how it signals and what we know about it. So the G-alpha-Q subunit is a heterotrimeric G protein, and in the resting state, so when the G protein couple receptor does not receive signal from a ligand, the alpha subunit is bound to the beta and gamma, and this trimeric G protein is bound to GDP, and so this maintains the resting state. when a ligand binds the G protein couple receptor, the G alpha subunit dissociate from the beta and gamma, and GDP gets converted to GTP. This allows for signaling. And what we know about the GNAQ Q209L mutation is, first of all, computational studies have shown that the result of the conformational change of this mutation induces impaired binding of the G alpha subunit with beta and gamma. And this means that basically the G alpha Q subunit has difficulties in going back to the resting state to bind to the beta and gamma. And also with this, there is impaired hydrolysis of GTP to GDP. And so resulting in a constitutive binding to GTP that allows for signaling for downstream pathways. And the downstream pathways that have been most studied in relationship to this mutation are the PLC beta and MAP kinase pathway and the trio RORAC and YAP pathways, which have been discovered in association with studies in uveal melanoma done by Boris Bastian and Silvio Gutkin. So I want to mention that this mutation, but also the 183, have been found in uveal melanoma, which is a very aggressive tumor of the eye. And this image on the right shows that if you express in mouse melanocytes the mutant form of GNA11 or GNAQ, and again, these are highly homologous. They share about a 90% sequence homology. You see increased phospho-ERK, so activation on the MAP kinase pathway. And this hyperactivation is similar to oncogenic drivers such as BRAF V600E or NRAS G12V. And work by Silvio Gutkin also showed hyperactivation of the YAP signaling pathway. But what we don't know is, are those pathways really the same pathways hyperactive when the mutation is expressed in endothelial cells? And how, can we make a model of GNAQ-related vascular tumors? Can we start investigating what happens? And the research question that we had was really, how is the endothelial specific expression of GNAQ Q209L driving vascular tumor formation? Is it using the same pathways identified in uveal melanoma or different kinds? And to do so, Sandra Schrenk, a very talented senior postdoc in my lab, has really spearheaded this project. And the way we start, I mean, it's a long story short. We did a bunch of work beforehand before coming to this, but nonetheless, this is the most impressive results that we got was when we bred mouse expressing the mutant allele GNAQ-Q209L under the ROSA26 locus, and it's a floxed allele. So when recombination with when crossed with the mouse expressing a CRE-RT2 recombinase under the CDH5 CRE promoter, so we can introduce expression of the mutant allele specifically in endothelial cells upon tamoxifen administration, and resulting in basically specific expression of the mutant GNAQ in the endothelial cells when we decide to administer tamoxifen. So in this scheme, we use adult mice. We've used also pups, but in this talk, I'm not going to go talk about them, but you can find that information also in our published studies. Here, I want to talk about adult mice because that's what allows us also to do more therapeutic studies and proof of concept studies. And when we administer tamoxifen, we do 2 pulses at 75 milligrams per kilogram. Mice, mutant mice administered with tamoxifen die pretty quickly. And we have chosen day 6 for our analysis to at least catch 50% of the mutant mice before they die. And you can see that different control genotype here, actually, they can live very long. Even the mutant mice without tamoxifen can live very long. So the phenotype is specific to the mutant genotype with tamoxifen. And we've analyzed, first of all, the subcutaneous tissue where most of these vascular tumors really appear. And on the top, you can see the control subcutaneous tissue. And on the bottom, the mutant mice. You can see the blood vessels are grossly enlarged. And we can see those red areas, which pointed us to think about vascular tufts, which is a very important histopathological feature of these vascular tumors with GNAQ mutations. And we thought these could be little hemorrhagic areas, or they could be those vascular tufts. And when we stain the muscle underneath the subcutaneous tissue in the chest with CD31 to label the vasculature, you can see there are enlarged blood vessels. And we also analyzed the vessel size distribution. And in the mutant mice, the percentage of large blood vessels with an area above 100 micrometer square was increased compared to the control mice. And also we quantified vascular density, the number of blood vessels per millimeter square, and the vascular area in percentage, and those are increased in the mutant mice subcutaneous tissue. We went on to also study the intestinal tissue because there are reports of KHE being also localized in the intestine. And so here you can see a gross image of the intestine. And again, you can see how much more prominent the blood vessels are. And again, you can see those vascular tufts. Whole mount immunostaining for CD31 highlighted again that the blood vessels are enlarged, and those focal enlargements that we call vascular tufts are very evident here as well. We next wanted to know if those vascular lesions were proliferative, and that's what, if hyperproliferation was really what was driving enlargement of these blood vessels. And to do so, we administer mice control and mutant with EDU 24 hours before analysis. Here you can see that in control mice on the left, in maroon, there is labeling for CD31, and then in green, the EDU positive nuclei. Then most of the proliferative cells, labeling EDU in green, are localized outside of the blood vessels in those adult control mice. Conversely, in the mutant, you can see that many of the proliferating EDU positive cells are localized within those enlarged blood vessels. And here is a quantification of the phenotype, and we quantified specifically the EDU positive endothelial cells. Next, we really wanted to dig deeper into the pathways upregulated or downregulated in the GNAQ mutant endothelial cells. To do so, we went back to an in vitro system, and we used the HUVEC or endothelial progenitor, endothelial colony forming cells, and we transduced them with a lentivirus expressing a doxycycline-inducible form of GNAQ wild type as a control, or GNAQ Q209L. So these cells prior to doxycycline administration are normal, and then when we add doxycycline to the medium of the cell culture, then they can be induced to overexpress the GNAQ wild type, or Q209L. And we did performed RNA sequencing. And what I want to show you first is the results from the principal component analysis that shows that the wild type cells without DOX or plus DOX cluster very similarly. Conversely, the cells with expressing GNAQ Q209L plus doxycycline cluster very, they are very separate. And so meaning that there are true differences that are due not only to the specific expression of the mutation and not the overexpression of the gene. So at least highly suggestive of that. The results for from the RNA sequencing highlighted important pathways. And one of them was the MAP kinase signaling pathway, complement and coagulation cascade. Very interestingly, it's not highlighted here, but a good internal control. We also saw G protein caporeceptor signaling pathway, inflammatory response, and angiogenesis. On the bottom, you can see how we confirm by qPCR also the upregulation of those or a differential expression of these important regulators. And here I want to spend some time talking about the coagulation, which was sort of something we weren't counting on, but sort of made sense to us. So an important complication of caposiform hemangiothelioma and tufted angioma is this coagulopathy termed Casabat-Merritt phenomenon by the two investigators who first described it in 1940. At that time, there was no biorender, and investigators had to draw also their images in papers. So we are lucky nowadays. At least I'm very bad at making drawings. So, and again, this is a consumptive coagulopathy that in the 30% of cases can lead to death. And it's associated with about 80% cases of KHE and 30% of cases of tufted angioma. And it's diagnosed in patients with thrombocytopenia, low platelet number, anemia, elevated D-dimer, and low fibrinogen. Surgery is not recommended because the number of platelets in the circulation is low, and so there is high risk of bleeding. And platelet transfusion should be avoided because what happens in Casabat-Merritt phenomenon is that the platelets get sort of trapped and stick to the malformed blood vessels. So if you add platelet, they will all go in the malformed region and worsen the disease. And importantly, really, even the idea that the platelets accumulate in this area is well known in the clinical world. Everybody talks about this, but I've looked extensively in the literature and I could not find a single image of a tissue section where you could see this phenomenon. And so really we don't have documentation of this, at least to my, basic science knowledge of looking for papers and cases. And we really don't know what are the molecular drivers of this. And there's no animal model to study it. So we wanted to know, based on the RNA sequencing data, suggestive that endothelial cells are already prone to maybe be pro-thrombogenic. We wanted to look at what was happening in the mice. And so we collected blood. And we could, in fact, see that the mice had thrombocytopenia, low platelet number. They had anemia and elevated D-dimer, suggestive that these mice were developing coagulopathy, had developed a coagulopathy, which we can call it KMP-like at this point. And then we also wanted to really document, are the platelets really accumulating in these malformed vessels? And is it because of this that we have less platelets in the peripheral circulation? To do that, first we perfuse the mice. So we infuse them with saline solution to wash away all the blood from the blood vessels. And we stain for a whole mount, the intestinal muscularis, for CD41 to label platelets, TER119 to label erythrocytes, and in green for CD31 to highlight the blood vessels. You can see a 3D reconstruction. Effectively here, the perfusion washed away all the blood. We don't see any TER119 or CD41's positive signal. But in the mutant blood vessels, you can see that even if we perfuse, these platelet and erythrocytes stick, especially in the tuft area, in those enlarged malformed vessels. We also really wanted to have a system, in addition to the in vivo, a simplified system that could test our hypothesis that the mutant endothelial cells were really the driver, or they contributed significantly, so they are sufficient to induce this KMP-like phenotype. And so in collaboration with Riccardo Barile here at Cincinnati Children's, we developed this blood vessel on chip. And so you can see we generate in this material an inlet where we can see the endothelial cells. And then we can perfuse these channels with blood. And we use controlled donor blood, human blood. And so what we did, we used the cells that I've described you before. We seed them in these channels with these inlets. And then only after they've attached and they're doing well, then we add doxycycline for 24 or 48 hours to induce expression of the mutant GNAQ. This is how the cells look like before perfusion. We stain for PKM1 or CD31, so you can see how the cells look like. And then basically, when we add the blood for the perfusion, we also add antibodies that can label fibrin deposition or platelets. And you can see that in the mutant cells in the bottom, when we perfuse with blood, fibrin deposits accumulates much more than you can see in the wild type, and really also acquires this more fibrillar structure. And also there is increased deposition of platelets, as you can see also in those quantifications. And so this is really suggestive that endothelial cells with the mutation by themselves can sort of drive the phenotype. Although we cannot exclude that there are secondary players in all of this that might exacerbate the phenotype. And the next, I want to move on to tell you about some of the other pathways that we identified thanks to the RNA sequencing data. And namely, we were really interested in the MAP kinase signaling pathway because it's, it's kind of trendy in vascular anomalies to look at it nowadays. There are inhibitors. And also we knew, this was a putative, important signaling cascade that could have been activated downstream of the mutant GNAQ. And so what we wanted to do was to confirm that this was really dependent on the mutation and how much is expressed. And Lindsay Bishop, a very talented grad student in the lab, helped with these experiments where we took the wild type expressing endothelial cells and the mutant GNAQ expressing cells, and we administer increasing doses of doxycycline. You can see that the expression of GNAQ is dependent on the dose of doxycycline that we administer. But then in the wild type, phospho-ERK remains the same. But in the mutant cells, you can see there is an exquisite increase in phospho-ERK signal that is dose dependent, depending on how much doxycycline we give. And we don't see any increased phospho-AKT. And here on the right, you can see the quantification of this data. We also wanted to confirm that this was not just something that we could see in vitro and in our mouse model, but we really wanted to go back to the patient tissue. And so we had a vascular tumor with this GNAQ Q209L mutation, and we stained first with UEA or Ulex europaeus agglutinin 1, which is a marker that labels selecting that can attack and label specifically human endothelial cells. And you can see those vascular tufts, these clusters of hyperproliferative endothelial cells and enlarged blood vessels. And when we stain for phospho-ERK, you can see there is increased signal in the vascular tumor. And then we double label with phospho-ERK and UEA1 and quantify the number of cells that are double positive for phospho-ERK and endothelial cell marker. And you can see that this number is increased compared to foreskin tissue from 5 different donors. Strong of this data, we went on and thought about, okay, if we inhibit MEK, can we prevent the phenotype? And this was a first proof of concept experiment, which is a preventative study where the administration of trametinib and control vehicle starts from the same day where we administer tamoxifen. And what you can see here, you will see only mutant mice were treated with vehicle or trametinib in the subcutaneous tissue. The blood vessels look normal. We don't see much of a phenotype. And upon quantification, you can see that the vascular density and vascular area are now down to the levels of control, non-mutant, and challenge mice. And the same thing was true for the intestinal muscularis tissue, where we don't see those vascular tufts in trametinib-treated mice, and the blood vessels now look more normal. And again, quantifications for vessel diameter and vascular area show numbers in the trametinib-treated mice that are similar to control and challenge mice. How is this happening? So we look at proliferation, and in fact, we see that trametinib treatment prevent this hyperproliferation, as you can see in the quantification. And we also look at the coagulopathy, and we can see that we also prevented the onset of coagulopathy with this pre-treatment or preventative treatment with trametinib. So we see platelet number and D-dimer are now similar to control and challenge mice. But this was sort of a proof of concept experiment, and we really wanted to know what happens if we start treating mice that already have preformed lesions. To do that, we had to change our tamoxifen dose. We went down to a lower amount, and now mice live at least up to day 20. And in this, with this scheme, now we did tamoxifen administration, and then started trametinib treatment or control vehicle at day 8 after tamoxifen. And we know that at this time point, there are already visible lesions when you open up the mouse. And so you can see that compare in red, those are the mutant vehicle treated mice. So they die pretty, within sort of the time frame that I've shown you. But when you treat with trametinib, we significantly extended the lifespan of these mice. We went back also to perform RNA sequencing to understand what changes trametinib treatment was responsible for. And especially, so here I'm showing you that when you look at the genes that are upregulated in the Q209L GNAQ cells compared to the wild type, we identify 617 genes being upregulated. And trametinib downregulates 73 of them. To show visibly in a heat map, you can see, for example, those are the GNAQ wild type cells, and we have increase of regulators of MAP kinase activity, inflammatory response, and so many of those then get normalized by trametinib. But it might interest you that while a good number of important genes go back down to normal, we still have a high number of genes that are still upregulated. And in fact, our mice still went on to die. So, and now we are really focusing on this gene and trying to identify important signaling pathways that we can think of targeting, possibly in combination with trametinib. And we have our eyes on the JAK-STAT pathway and NF-kappa B. And so more to come in the future. At this point, I don't have anything to show you about that. But here I want to summarize what I've shown you today. So we talked about vascular tumors with mutations in this GNAQ, and namely this GNAQ Q29L. And this induces hyperactivation of at least one of the pathways, the PLC-beta MAP kinase ERK pathway, leading to formation of vascular tufts and coagulopathy. And trametinib can prevent this phenotype and extend the lifespan of mice. I have a few more slides to show you. So couple of things we're interested on. First, we wanted to better recapitulate the somatic nature of those GNAQ mutations. And the way we've done this before, we just gave tamoxifen, and we gave maybe, lower doses to think maybe we can hit less endothelial cells and create more of a mosaic. And now what we are trying to do is to give topical 4-OHT tamoxifen only in the ear. So we do know that even if you give it only in the ear, some of it will go systemic. So just want to let you know to the field that it is going to go systemic. If you have a strong phenotype, your mice will still end up dying. But you can focus on analyzing the phenotype because it will be stronger in that area. And so what you can do here is in these beautiful images, we can look at the same ear at different time points and really do a longitudinal study with this. And we think that's the sort of the beauty of this, because when you do systemic, lesions can happen here or there, and you might not know where to look at. And unfortunately, we don't have an organ-specific or tissue-specific endothelial Cre driver yet. So this, I think, is the best we can do these days. But you can see in the mutant mice, when you add tamoxifen in the ear topically, you can see from day 0 to 25 that the blood vessels become much more prominent and enlarged. And this is a control mouse, control genotype treated with tamoxifen, nothing happens. And we wanted to use this system to investigate the effect of trametinib in a sort of a, system where we could treat for longer, and also start investigating the effect of rapamycin, because there are reports out there that show that rapamycin is somewhat has a good level of efficacy in patients with caposiform hemangiopericytoma. So we wanted to know, is this because these patients maybe have a different mutation, or is this going to work even with this GNAQ Q29L mutation? And if so, how does it work? And so here you can see I just want to specify that while the tamoxifen is given only in the ear, the trametinib or rapamycin treatment is systemic at this point. We want to repeat and do it also topical, but for now we just did systemic. And you can see trametinib work pretty efficiently also in this system. And rapamycin is not, we don't think it's as good, but we have a lot of ears to analyze, so I don't have the full quantifications for you, but I really wanted to share this system with all of you guys and this preliminary data. But I think it is working to some extent, and the combination so far we can't really tell. But stay tuned for all of these quantifications and more data, and we are really interested that rapamycin is working and trying to understand later on how at the molecular level. And next, I really want to spend just a couple of minutes in highlighting also the intestinal muscularis that I've shown you today throughout the slides that we've been using this system. And I kind of want to share this system with the vascular community out there, because we think it's a very good system. And Sandra Schenk in my lab really sort of devised this and made it as a beautiful tool to look at blood vessels. And so what we're looking at specifically is this specific layer of the intestine, which is called muscularis propria. And this is very finely vascularized. You can see how beautiful here in the CD31 staining the vascular plexus is. And it's also fairly simple. But the advantages of this system is that we don't have to do any tissue clearing, because we sort of peel off the layers and make it to a thin, and we virtually see any background with all the stainings that we've done so far. And the intestine is very long, so you can get a lot of tissue, you can fix it and go back to it later on and flat mount it and look it in a slide. So we think this is a very good system. So we're trying to write up little protocol manuscript, but here I am saying, if you want to use it, please contact us, and we're happy to share before we're going to be able to publish. And I just want to quickly tell you that with this system, then you can do EDU labeling and measure proliferation. You can use the Nikon elements to basically mask and count only the proliferative cells within the CD31 blood vessels. And this allows us to see, Lindsay Bishop has done this quantification and show that, for example, in young animals at 2 weeks, there is high proliferation in the blood vessels, but also in the surrounding tissue. And in adult mice, the muscularis is pretty much not proliferative. You see a couple of nuclei here, but we have much less proliferation. We can also do phenotypical analysis and use IMAGE to measure the vessel diameter, or also use Angio tool to measure or quantify vascular area, diameter, or branching points. And last, You can also do your favorite staining. You can also stain for live one. Here I want to just say disclaimer, lymphatics are present only in the colon, but in the muscularis of the small intestine, it's not seen. So, and then you can study the enteric nervous system and stain for nerves with beta-3 tubulin, or stain for erythrocytes and platelets, or for your other favorite cell type. And with this, I want to just make one announcement that we're looking for method papers. If you have your favorite in vitro, three-dimensional, or in vivo model, there is patient-derived or patient-related. So it can be even a mouse model with a mutation that is found in patients. We would love to have a collection all together. And if you are unsure, you can contact me so we can revise your abstract. Looking forward for your submissions. And we've published with them before and had a pretty good experience. So we've enjoyed, it really makes the team to work together, to have the video put together. It was a very lovely experience for us. So that's why we accepted to also organize this collection. And last but not least, I do want to thank people who did the hard work. And so first of all, Sandra Schrenk, she's done phenomenal work. Lindsay Bishop is a grad student in the lab. And then also Jo Regatz is a hem-onc fellow in the lab, and she's helped with some of the coagulopathy studies. I want to thank collaborators at Children's and outside that share mouse lines. I want to thank funding sources, and we're looking for a postdoc. So this GNAQ project, we have many directions that we want to take to look at molecular regulators of the coagulopathy and find additional targets. And we really got recently a very good score in a NHLBI grant. So we're expecting good news and need people to work on it. And thank you so much. I'm happy to take any question at this point. Thank you.
Great presentation, really exciting work, and not only the mouse model, but also the in vitro system that you presented with the where you were using blood to study this coagulopathy. I'll start the discussion.
Thank you.
Really interesting mouse model with the expression of GNAQ throughout the endothelium. I was curious, did you see any sort of differences in sensitivity to the mutation between arteries, capillaries, and veins, or did it were all affected by the mutation?
So yes, this is a very good question, and one that the field is very interested to address. Initial studies we have done also in the retina, and where it's a very beautiful system where you don't really need to mark, but you can see which ones are the arteries and veins. And we saw that there is a exquisite sensitivity in the capillary venous plexus, and we never see those enlargement in arteries. Disclaimer, so in the intestinal muscularis, it is a beautiful system, but we don't see big vessels, arteries there. Mm-hmm. So we can't say there, but at least in the retina model, this is suggestive that arteries are not affected.
Right.
You might want to know about lymphatics, and I think this question is coming. And again, we look specifically in the small intestine, so where there are no lymphatics. Yeah. We now realize, hey, in the colon there are lymphatics, so maybe we should look there. Yeah, I don't have a good answer yet.
And we're more than happy to share our ear skin whole mount staining protocol with you that we use for lymphatics and blood vessels.
Yeah, and to this extent, I think I do want to know which one would be, if we were going to use a lymphatic specific redriver, which one the field would suggest us to use to see if we put the mutation specifically. Because at this point, if we see defects in the lymphatic vessels, also we couldn't be sure that they are primary defects or secondary, because the blood vessels themselves are so abnormal that it could be a secondary defect. So
there's a PROX1 tamoxifen inducible Cree at JAX, which is a live colony, and I think that's a great one. There's also VEGF receptor 3 pre-ERT2. The challenge with that is that there are some blood vessels that express VEGF receptor 3. So if you give it systemically, that could be an issue. But topically, especially for the ear, it's very specific to lymphatics.
Okay, we might start doing that we've always, also in KHE, there are some lymphatic vessels that are affected. So, and again, the question is it a primary defect, secondary defect, or the mutation happens in a progenitor? Yeah.
We have a question from Joyce.
Good.
I can read it, or Joyce, do you want to unmute yourself and ask?
Yeah, Elisa, can you comment if you think there's any crosstalk between the phospho-ERK and AKT? Because on your Western blots, it looked like as in the mutant cells, as the phospho-ERK went up, AKT went down, and then that pathway came up in your trametinib treated. Yeah, so I'm just, can you have any thoughts on that?
It is interesting. We haven't dig too deep yet. Yes, it's true that when phospho-ERK is going up, phospho-AKT seems to go down. And in the RNA sequencing, I think there are some problems, how people cluster this stuff. We see genes like VEGF receptor 2, and, it's how, those pools of pathways, the genes that they put in these pathways might not necessarily, even they are clustered into this PI3 kinase AKT pathway, they might not all be related, because in fact, then we don't see increased phospho-AKT. In response to trametinib treatment, we didn't look too carefully if AKT goes high when you suppress phospho-ERK, so I don't have a good answer for that. What we do see is increased phospho-S6.
Oh, okay.
That's how rapamycin might be effective there, but it might be stimulated by increased phospho-ERK. So we haven't, it's very interesting, but we haven't had time to go into that yet. Yeah, it's a very good question.
Okay, thanks.
Hi, Lisa. It was such a great talk. Yes, we're homesick today. But I was wondering if you looked at any other vascular beds besides sort of what you presented today, for example, either in other organs like the liver and the brain. And I was also wondering if you ended up seeing any hemorrhages in any places.
Very good questions. So we did look, for example, we look in the retina and hyaloid, and we do see abnormalities. We've looked in the brain, and we do see abnormal vessels. I believe we tried to look in the lung, but couldn't, I don't know, we just haven't done good enough. In the heart also, we see some kind of abnormalities in the vessels. And I don't know what else we looked at. I don't think we've looked too carefully in the liver. And then your second question was
about if you saw any bleeding in any
yes, yes.
With the anemia, if it's just due to the coagulopathy, if there's maybe a bleeding issue as well.
Yes, we do see a bleeding issue. So we have done even, we didn't even need to do dextran to see extravasation. We just did, I didn't put this data out there today, but we saw that the erythrocytes can really extravasate outside of the blood vessels. And then we followed up with in vitro studies, and we do see that the V-cadherin at the junctions is reduced in the mutant cells. So there definitely is bleeding. Yeah.
Thank you.
Seeing the special guest with Sarah just reminded me something. We're going to be announcing a Young Investigator Awards soon. So we'll be sending that announcement out, I think, next month. I don't think there are any additional questions, but I just want to thank you once again for a wonderful presentation, and thank you all for attending and participating.
Thank you so much.